Hello everyone. Efruzhu Cancer Carcinogenesis Abstract Book Volume 3 A.B. Efruz Asiloğlu Cancer Theoretical Independent Researcher. Two of our most important antioxidant enzyme, copper zinc superoxide dismutase and ceruloplasmin. Copper deficiency can significantly increase the plasma cholesterol concentration, results in an increase in low-density lipoprotein cholesterol and a decrease in high-density lipoprotein cholesterol. The functioning of macrophages were decreased in severely copper-deficient rats. Most tumor cells have decreased copper-zinc superoxide dismutase activity compared to normal cells, and copper salicylate have been shown to possess anti-cancer, anti-carcinogenic, and anti-mutagenic effects. Copper-containing enzyme ceruloplasmin is significantly elevated in inflammatory conditions and has anti-inflammatory activity. Copper excess transport protein ceruloplasmin, copper albumin, also exist in unbound form known as copper ions. These ions can be powerful generators of free radicals. Copper is involved in carcinogenesis not by direct mechanism but by antagonizing selenium, thereby diminishing selenium protective effects against cancer. Zinc and vitamin C supplements are strong antagonists of copper status, free radicals, and inflammation, two well-known cancer inducers. Copper serum levels are elevated up to threefold above normal with inflammations and with many chronic and infectious diseases. The most effective anti-inflammatory agents are copper complexes. Commonly, these are related to salicylic acid. Copper salicylates have a similar action as superoxide dismutase to protect cells from free radicals. Thioredoxin can promote B-cell differentiation. Thioredoxin efficiently increases intracellular calcium and activates protein kinase C through its translocation to the membrane. Thioredoxin functions in the activation, differentiation, and proliferation of lymphocytes. All cancers exhibit a common phenomenon: the massive arrival of iron and copper at the tumor site, rusting agents, oxidants. Phagocytes over-respond to the cellular chaos and arrive in such large numbers at the tumor site. They also begin to dump iron and copper. The result is destruction of the surrounding tissues and eventually loss of function of the affected organ. Iron feeds tumors cells. Iron is growth factor for virus, bacteria, fungi, and tumor cells. Take away the iron and copper, then you stop tumor growth and shrink the tumor. Aspirin and other non-steroidal anti-inflammatory drugs reduce the risk of cancer by inducing apoptotic cell death. A critical pathway for apoptosis involves the release of cytochrome C from mitochondria. This interacts with APAF1 to activate caspase proteases that orchestrate cell death. Respiratory deficiencies common to rapidly growing cancer cells. Respiratory deficiencies might result in differentiation of cells and hence neoplastic transformation. Transformation was the result of submolecular microelectronic changes involving errant dismantling and rebuilding of the electron transport chain during cell division, and that mutation in mitochondria might cause cancers. Reduce pyruvate oxidation and increase lactic acid production, increase glutaminolytic activity, reduce fatty acid oxidation, increase glycerol and fatty acid turnover, modified amino acid metabolism, and increase pentose phosphate pathway activity. Analysis of the inner membrane lipid composition of various tumor mitochondria has indicated elevated level of cholesterol, varying total phospholipid content, and or changes in the amount of individual phospholipids relative to normal controls. Pathogenesis of prostate cancer involves the mitochondrial metabolic transformation of citrate-producing cells to citrate-oxidizing cells. This metabolic abnormality is not reported in the other cancers. The mitochondrial membrane potential of carcinoma cells is approximately 60 millivolt higher than that of control epithelial cells. Cancer cells have dysfunctional mitochondria which prevent their use of the citric acid cycle. Consequently, pyruvic acid, the normal end product of glycolysis, which normally would enter the mitochondria for its total combustion energy, is instead converted to lactic acid. Cancer cells can produce 40 times more lactic acid than normal cells. Cancer cells can't utilize the Krebs cycle as efficiently as normal cells, if at all. Consequently, they must convert pyruvic acid into lactic acid and must also increase the production and activities of their glycolytic enzyme in order to survive. The lactic acid so produced can then serve as a source of fuel energy by being carried to the liver, reconverted into glucose via the pathway of glycogen cycle, and finally returned to the cancer cells. Cancer cells have little or no superoxide dismutase in their mitochondria without adequate protection from SD superoxide, a normal toxic radical byproducts of the Krebs cycle of metabolism would injure the genes or proteins in the mitochondria. This would impair the function of the Krebs cycle and prevent the entry of pyruvic acid into the mitochondria. Consequently, pyruvic acid must be converted into lactic acid instead of its normal breakdown into carbon dioxide and water. Caloric restriction results in tumor shrinkage is that it contributes to the increase of ketones in the blood. This in turn inhibits the activity of phosphofructokinase, an enzyme that plays a key role in the regulation of glycolysis. That ketones can inhibit the function of phosphofructokinase. On a restricted caloric intake, especially one reduced by 40%, the body must burn its own fat as a source of fuel energy. Fats are converted into ketones by the liver and then deposited into the blood for distribution to cells throughout the body. Normal cells can burn fats and ketones in their Krebs cycle and can survive without glycolysis. Citric acid, an intermediary product of the Krebs cycle metabolism, has also been reported to block the action of phosphofructokinase. A low carbohydrate, high fat diet to increase the blood level of ketones. Triiodothyronine T3 is considered a major regulator of mitochondrial activity. The more delayed influence of thyroid hormone upon mitochondrial respiration probably result from mechanism elucidated at the nuclear level, including changes in phospholipid turnover and stimulation of uncoupling protein expression, leading to an increased inner membrane proton leak. A43 KDACERB and A alpha 1 protein located in the mitochondrial matrix P43, acting as a potent T3-dependent transcription factor of the mitochondrial genome, induce early stimulation of organelle transcription. T3 increases mitochondrial TFA expression, a mitochondrial transcription factor encoded by a nuclear gene. Direct mitochondrial T3 pathway involving P43 led to the conclusion that it is not only involved in the regulation of fuel metabolism but also in the regulation of cell differentiation, as the processes leading to or resulting from differentiation are energy consuming. P43 coordination of metabolism and differentiation could be of significant importance in the regulation of development. Excessive unsaturated fats inhibit all body systems mainly by inhibit enzyme essential to metabolic processes required for health and immune protection, unsaturated oils inhibit proteolytic enzymes, and this has far-reaching effects. Inhibition of proteolytic enzymes by unsaturated fats causes trouble at many sites where proteolytic enzymes are necessary. Active thyroid hormone and the digestion of cellular proteins involved in maintaining a steady state as new proteins are formed in the cell. There is an enzyme system called the protein kinase C, PKC system that is excessively activated by certain substances and certain conditions. Substances that cause excess activation of this system are polyunsaturated fatty acids, including free linoleic and linolenic acid, excess estrogen, a cancer promoter and cancer promoting phospholiesterase. These substances stimulate the cell while blocking the energy it needs to respond. The protein kinase system is also abnormally activated in diabetes and cancers. Unsaturated fats cause thyroid suppression and lead to hormonal imbalances. Unsaturated oils cause blocked thyroid hormone secretion, its circulation, and its tissue response. This leads to increased estrogen levels. Thyroid hormone is essential for making the anti-aging hormones, namely pregnenolone, progesterone, and DHEA. When your thyroid is in trouble, the manufacturer of these anti-aging steroids are in trouble. Thyroid converts cholesterol in your body to those anti-aging steroids. Low thyroid function can lead to high cholesterol. Unsaturated oils inhibit cellular respiration. Mitochondria contain some unsaturated fats to allow them to take up water. Stress and hypoxia, oxygen deprivation, cause cells to take up large amount of unsaturated fatty acid where they can directly damage mitochondria. Cancer cells are known to have a high level of unsaturated fats. Increased circulating polyunsaturated fatty acids can block the Krebs cycle. Polyunsaturated fatty acids and estrogen act similarly on time. Same regulatory pathway is important. Both inhibit thyroid function, inhibit vitamin E. Polyunsaturated fatty acid and estrogen in reproduction may be similar, namely the promotion of cell division, essential for reproduction but dangerous in abnormal cell division such as cancer. Stress and hypoxia, oxygen deprivation, can cause cells to absorb large amount of unsaturated fatty acids. It is well known that cancer cells are dependent on unsaturated fatty acids for life and growth. Unsaturated fats are essential for cancer. At least part of the effect of unsaturated oils is to inhibit thyroid function. Inhibiting the thyroid gland unsaturated fats impair intercellular communication, suppress several immune functions related to cancer, and are present at high concentrations in cancer cells where their anti-proteolytic actions would be expected to interfere with the proteolytic enzymes and to shift the equilibrium toward growth. Release cytochrome C playing role in the cell defense mechanism against neurotoxicity. Process of neuronal death is called excitotoxicity and appears to involve sustained elevations of intracellular calcium levels, impairment of neuronal energy metabolism, may sensitive neurons to excitotoxic cell death. Neurons constantly require new carbon skeletons glial to sustain their TCA cycle, tricarboxylic acid cycle. When these supplies are withdrawn, neurons are unable to generate amino acid transmitters, and their rate of oxidative metabolism is impaired. Radicals, reactive oxygen species, have both beneficial and harmful actions in biological tissues. They are known to have a crucial role in stimulation of phagocytosis, induction of drug detoxification pathways, and stimulation of signal transduction pathways. Isoprostanes are prostaglandin-like compounds that are produced by free radical-mediated peroxidation of polyunsaturated fatty acid. High oxidative stress brought on by over-exercising and probably high caloric intake. Some marathon runners are burnt out in their early 40s, resulting in an overall lower amount of energy. Feedback mechanism under high oxidative stress would be working overtime to produce at a normal gene level increased amounts of enzymatic quenchers, antioxidants produced in the body, such as superoxide dismutase, catalase, peroxidase, etc. Caloric restriction. We know if we decrease calories by 20%, animals live 20% to 30% longer. With lower caloric intake, there is lower metabolic activity and less free radical production with lifespan. Endothelium derived nitric oxide. Endothelium derived nitric oxide. Cerulenin, a fungal metabolite, is known to be a specific inhibitor fatty acid synthase. Cerulenin is an effective inducer of apoptosis in the different wild type p53 and mutant p53 tumor cell lines. Cerulenin treatment caused a rapid release of cytochrome C from the mitochondrial intramembrane space into the cytosol, followed by activation of caspases 9 and 3. The breast carcinoma cell line SKBR3 exhibited significantly higher fatty acid synthase expression and activity than any other cell line. Manganese superoxide dismutase, antioxidant protein, produces the oxidant hydrogen peroxide. Vitamin C is known to be a DNA protecting antioxidant, is a switch hitter, also capable of inducing the production of DNA damaging compounds. They begin by converting linoleic acid, the major polyunsaturated fatty acid in human plasma, into another compound called lipid hydroperoxide. When certain metal ions are present to act as catalysts, the lipid hydroperoxides degrade further into DNA damaging agents called genotoxins. These compounds react with DNA switching one base for another in mutations that have been found in human tumors. Vitamin C might also be capable of making lipid hydroperoxides degrade into genotoxins in place of the transition metal ions. Vitamin C produces significant amount of genotoxin in test cell, and whether they generate cancer-causing mutations. Free radicals can do a lot of damage by reprogramming our DNA. Nuclear encoded respiratory chain proteins remain primarily unaffected, indicating that this differential affection of respiratory chain subunits can importantly contribute to the induction of programmed cell death in aging and or calcium overloaded cardiomyocytes. Accumulation of damage to the mitochondrial DNA should consequently lead to a defective synthesis of mitochondrial genome-dependent proteins of the respiratory chain, with subsequent impaired function of the complexes containing these proteins, but no impairment in the function of complex 2. There are three expected consequences resulting from a defective respiratory chain. At first, the mitochondrial energy output could be diminished due to impaired proton pumping. Secondly, impaired pumping of protons through a defective respiratory chain from the matrix to the intermembrane space causes an increased generation of reactive oxygen species in the mitochondrial matrix. Finally, an enhanced mitochondrial radical formation should cause an unstable or decreased mitochondrial transmembrane potential. During normal respiratory conditions, 1-3% of the electron flux is continuously converted to free radicals at several sides of the respiratory chain. High cellular oxygen concentration or reduced availability of reduced cofactor of the respiratory chain and the high transmembrane potential of the inner mitochondrial membrane tend to enhance this mitochondrial radical formation, which is substantially enhanced in presence of defects or imbalances within the respiratory chain. Potential mechanism for the shift in substrate utilization and oxidation include changes in mitochondrial lipid content, lipid composition, and protein interactions, as well as oxygen-free radical injury with subsequent lipid peroxidation of mitochondrial membranes, leading to significant membrane dysfunction. Oxygen-free radical injury to key enzyme system for fatty acid oxidation may play a role. Another potential mechanism includes changes in nitric oxide levels with age. Recently, it has become apparent that nitric oxide produced from endothelial nitric oxide synthase plays an important role in the regulation of myocardial substrate metabolism. It has been shown that cardiac nitric oxide production inhibits myocardial glucose utilization. As a consequence, myocardial fatigue utilization is increased. The prostaglandin synthesis inhibitor indomethacin enhance the endothelin-1 induced vasoconstriction in 2 and 6 MO rats to a similar extent, was associated with the increased level of respective messenger RNA. Several lines of evidence indicate that nitric oxide regulates myocardial glucose metabolism. Cardiac nitric oxide production regulates myocardial glucose uptake via a cyclic guanosine monophosphate dependent mechanism and strongly suggests that ECNOS, endothelial nitric oxide synthase. In the regulation, this oxidative stress-induced activity promotes the production of number of pro-inflammatory cytokines, which can contribute to the pathology of many disease states associated with aging. Rate of glucose utilization were increased and rates of ketone body utilization were decreased in hypertensive health. The circulation of reactive oxygen species leads to the activation of reactive nitrogen compounds, which in turn induce or activate genes in the DNA that are associated with many degenerative diseases. The presence of white blood cells, they can produce harmful substances. In disease, energy is not put to useful work but released instead as heat. Examples of adverse heat production in biological systems are inflammation associated with arthritis and fever stimulated by infection. Maintenance of mitochondria requires not only adequate antioxidant but also precise regulation of intracellular calcium levels. Melatonin also may stimulate several antioxidative enzymes including superoxide dismutase, glutathione peroxidase, and glutathione reductase, as well as inhibiting the pro-oxidative enzyme nitric oxide synthase. Free radicals also increase production of vital hormones or chemical messengers like prostaglandins and cytokines. It can be seen that we need free radicals to maintain health. When the lining of the blood vessels is damaged, then one can see atherosclerosis develop. This is the method for the development of chronic disease. The immune system is one of the factors influencing susceptibility to cancer. Immune deficiency, there is an increased risk for cancer. Mitochondrial dysfunction causes cell damage and death by compromising ATP production, disrupting calcium homeostasis and increasing oxidative stress. Furthermore, mitochondrial damage can lead to apoptotic cell death by causing the release of cytochrome C and other pro-apoptotic factors into the cytoplasm. Oxidative stress is a particularly important factor in mitochondrial dysfunction because the respiratory chain continually leaks the free radical superoxide. Another radical that interact with mitochondrial metabolism is nitric oxide, which can react with superoxide to form the damaging byproduct peroxynitrite. Alpha-tocopherol in preventing atherosclerosis, inhibition of the protein kinase C activity by alpha-tocopherol was shown to be the basis of the vascular smooth muscle cell growth inhibition. The activity of protein kinase C by alpha-tocopherol results in different events in different cell types, including monocytes, macrophages, neutrophils, fibroblasts, and mesangial cells. Adhesion molecule expression and inflammatory cell cytokine production were also influenced by alpha-tocopherol. The important finding of the effect of alpha-tocopherol against atherosclerosis is not due only to the prevention of low-density lipoprotein oxidation, but also to the downregulation of the scavenger receptor CD36 and to the inhibition of protein kinase activity. Intracellular iron and calcium are important mediators of DNA damage. In response to oxidative stress, on the contrary, sustained generation of reactive oxygen species inhibited the execution of the apoptotic process. The selenium-containing compound epsilon has been shown to possess both antioxidant and anti-inflammatory properties. Signaling cascade regulated by tyrosine kinase phospholipase coupled activation of MAP kinases and protein kinase C in transmitting the death signal triggered by myocardial ischemia reperfusion. The study implicates that antioxidants generated as an important role in maintaining the balance between antioxidant reserve and reactive oxygen species, and thus dictates the need of antioxidant supplementation. Reactive oxygen species, either directly or via the formation of lipid peroxidation products such as acrolein, 4-hydroxy-2 nonenal and F2 isoprostanes, may play a role in enhancing the inflammation through the activation of stress kinases. Oxidative stress and pro-inflammatory mediators can alter nuclear histone acetylation, deacetylation, allowing access for transcription factor DNA binding, leading to enhanced pro-inflammatory tumor necrosis factor alpha, interleukin-1, interleukin-8, and protective antioxidant gamma GCS, manganese superoxide dismutase, heme oxygenase 1 gene expression, in various lung cells. The role of ascorbat on the nuclear binding of AP1 in macrophages. Ascorbat mediated regulation of AP1 binding was unrelated to free iron but may be regulated by new protein synthesis. Homocysteine accumulates in chronic renal patients due to both decreased clearance and impairment of renal metabolic function. Auto-oxidation of homocysteine generates reactive oxygen species, and this pro-oxidant nature of homocysteine causes oxidation of low-density lipoprotein, which contributes to atherogenesis. They reported that increased level of plasma homocysteine inhibits glutathione peroxidase activity in vitro of the enzyme. The findings suggest that hyperhomocysteinemia attenuates the antioxidant properties of glutathione and thereby potentiates peroxide-mediated cell injury and decreases endothelial cell messenger RNA expression. Nitric oxide is generated in the vascular compartment and plays a significant role in reperfusion injury by peroxynitrite formation. Oxidative DNA damage in streptozotocin indicated diabetic rats. The disruption of the balance between free radical production and cellular defense mechanisms as a consequence of ischemia, anoxia, and reoxidation injury have been implicated in a wide variety of acute and chronic neurologic disorders. Lipids are essential membrane constituents, but in addition, some participate in signaling cascades during normal cell function, while others are targets for lipid peroxidase during oxidative stress. DHA, docosahexaenoic acid, is an important membrane component which may accentuate the expression of various signaling cascades, including that of protein kinase C. The most frequent site for PGL is the carotid body. This highly vascular organ plays a key role in the acute response to low oxygen levels, hypoxia at the cellular level. This response involves activation of transcription factor, subsequent systemic response that includes an increase in red blood cell mass, stimulation of new blood vessel growth, and an increase in ventilation. Chronic exposure to low oxygen conditions, such as is experienced by those living at high altitudes or with certain medical conditions such as emphysema or cyanotic heart, induces cell proliferation in the carotid body. The hypothesis that the genetic defect in PGL might involve a critical component in an oxygen sensing and related signaling pathway. The mitochondrial electron transport chain might play a critical role in oxygen sensing and related signaling. The CYBS protein and the hypoxia associated with its Absence may play role in broad range of tumors. They know that many solid tumors are known to be hypoxic relative to normal tissue and that LOH of the region containing the SDHD gene is frequently seen in carcinomas of the bladder, breast, cervix, stomach, lung, ovary, and nasopharynx, and also in melanomas. Thank you very much.